So this talk is titled Population Genetics versus Human Evolution. So I want to read this quote to you. This is from a very famous former evolutionary biologist. Nothing in biology makes sense except in light of evolution. This is a common textbook quote you might find as a freshman biology student. However, famous evolutionary population geneticist Michael Lynch counters this, saying nothing in evolution makes sense except in the light of population genetics. That's a very important point here. This is a long quote, but I think it's important to read. He's a population geneticist, and he's criticizing people like Richard Dawkins, people who have made evolution very popular based on misconceptions about how evolution is supposed to work at the molecular level, at the genetic level. So he says, evolutionary biology is treated unlike any science by both academics and the general public. For the average person, evolution is equivalent to natural selection. And because the concept of selection is easy to grasp, a reasonable understanding of comparative biology is often taken to be a license for evolutionary speculation. It has long been known that natural selection is just one of several mechanisms of evolutionary change. But the myth that all of evolution can be explained by adaptation continues to be perpetuated by our continued homage to Darwin's treatise in the popular literature. For example, Dawkins, we all know who Richard Dawkins is, right? His agenda to spread the word on the awesome power of natural selection has been quite successful, but it has come at the expense of reference to any other mechanisms, a view that is in some way profoundly misleading. And here's another evolutionary population geneticist saying that no one can claim to understand how evolution works without some basic understanding of classical population genetics. Yet I have found, I say this humbly, most people, including scientists, including evolutionary scientists who are not in the field of population genetics, have a very overly simplified understanding of how evolution is supposed to work at the population genetic level. So what happened? At the time of Darwin, you're familiar with natural selection. He popularized it. He didn't invent that idea, he popularized it. But in the 1930s, it became clear, just before that, that how is Darwinian natural selection going to survive what we now know about Mendelian genetics? So in the 1930s, the founding fathers of population genetics, Ronald Fisher, J.B.S. Haldane, Sewall Wright, among others, they basically merged this Darwinian idea of natural selection with Mendelian genetics, giving rise to the Neo-Darwinian theory of evolution, which is the theory of evolution as we know it today. So that was a very important time in the development of the modern theory of evolution. And at its foundation is now population genetics. So what is population genetics? Population genetics is the study of changes in the distribution of alleles. Alleles are different variants of a gene. For example, when a mutation arises in a gene, it's now a different variant of that gene. So it's the study of the changes in the distribution of alleles in a certain population, considering the influence of the following evolutionary forces. I don't think these are evolutionary forces. We'll talk about that. But these are real processes. Natural selection. We all know what that is differential reproduction. Those with supposedly more favorable genotypes are more likely to pass on their DNA to their offspring. Genetic drift. This is where you have a more random fluctuation in allele frequencies, simply apart from natural selection. It's random. That's important. So when I have children, if I have a mutation in one of my chromosomes, I have two chromosomes, right? So for all of we have 23 pairs of chromosomes. So if a mutation arises in one of my chromosomes, there's a 50% chance I'll pass that on to my offspring. So right off the bat, you can randomly lose the new mutation right after it just enters the population. So those deviations from 50% chance of being passed on cause kind of like a random walk of changing allele frequencies. Mutation. We know what mutations are. Random copying errors in the DNA. Gene flows. This is where you can have genotypes enter or leave populations through immigration or emigration. Main point here is this. The main goals of this field of study is to determine how the frequency of a mutant gene will change over time under the influence of these evolutionary forces, to examine how genetic variability is maintained. In essence, distilling this to its simplest form, The real focal point is mutations. It's mutation accumulation in a population over time and all the factors that influence that process. And he adds one key point to this quote that I want to read. And this is the key. He says, and to establish the probability that a new mutant completely replaces existing variants of genes, a phenomenon called fixation. And I want to talk about genetic fixation. What is that? And why is that so fundamental to this process of evolution? And it's one of the cornerstones of population genetics. Yet I've found many people who believe in evolution don't even know what genetic fixation is. I don't mean that to be critical, it's just the truth. Most people do not understand these very fundamental concepts, and yet they can have such confidence that evolution is true. And you have to ask, how can you claim that if you don't understand these very fundamental concepts? So, how does evolution work? The popular perception of how evolution works is fairly intuitive and simple. And it's because of that I think it has such persuasive power. But you have to account for all the other factors. So the popular perception is that you have mutation plus some natural selection. So let's say you have a beneficial mutation, and that might be amplified in the population because it increases your chance of reproducing. And so they say, well, you just need a few beneficial mutations. Let's get rid of some of the harmful mutations by negative selection, and then we can evolve. And this has led to this idea of, well, evolution is intuitive. You just have a bunch of small little changes, microevolution, some might say, and those will eventually add up to big changes, macroevolution. They say, see, it's very intuitive. How can you deny it? So let me give you this example here. This is a population of, say, 9 individuals, 9 reproducing members of a population, and a mutation arises in a single individual. And so the smile face, they have a beneficial mutation, they have a reproductive advantage over the rest. In generation 2, let's say over time, this mutation increases in frequency in the population as a result. Now, you might say, based on textbook definitions of evolution, you'll say, see, that's evolution in action. We see a change in allele frequencies. There we go. That proves evolution is a fact. It's scientific. How can you deny it? We see this happening every generation. Therefore, these small changes in allele frequency can add up to big changes over time. You can begin to see how this oversimplification leads to claims like that. But this is incomplete. It's a gross oversimplification of how evolution is supposed to work at the molecular genetic level. So what's missing? The process of fixation is missing, among many other things. So what you really need is not only mutation and selection, but also fixation. Let me explain that. So here's the more complete process. You have to go from a rare mutant allele. It starts out in the population in one chromosome. It's very rare. It's vastly outnumbered by all the non-mutants. Then over generational time, it can eventually become fixed in the population. What does that mean? It means that rare mutant allele occupies both chromosomes in all individuals in the population. Then it becomes permanently established. Every reproducing member has both of those mutations in both chromosomes. Then it is permanently established, unless there's a back mutation, which is not likely to revert all the way back to this. But understand that it has to go from, this will make a lot of sense. Let's suppose you have a population of 10,000. And this is the effective population size. So these are the number of reproducing members of the population. Now, when the mutation first arises, it's vastly outnumbered by the non-mutant alleles. What's the frequency? Is it 1 out of 10,000 or 1 out of 20,000? Why is it 1 out of 20,000? Because we're diploids. We have 2 chromosomes, right? So it starts out 1 out of 20,000. To become fixed, it has to spread to all members of the population in both chromosomes. 20,000 out of 20,000 has to be the allele frequency. And that right there, from rare allele to fixed, represents just one step in evolutionary advance. That whole process. Because if it's not fixed, it can be lost from the population due to a process known as genetic drift. So this is a fundamental principle of population genetics, and that process has to happen over and over and over again to build genomes of new information, new genes, new novel functions and structures. So that's just one layer of realism that we're adding to the evolutionary theory. So here is from a population geneticist. Again, these are all so far from secular evolutionary population geneticists. He explains this process very simply. In population genetics, fixation occurs when every individual in a certain population has the same allele at a particular position on a chromosome. The allele, for example, a single mutation or a whole gene, will be rare in the beginning, but it can spread through the population by a process of random genetic drift or positive selection. We'll get into this more, but once the frequency of the allele is 100%, like I showed you, meaning that every individual of this population has this gene at a particular locus, then this gene is fixed in the population. This part is important too. The opposite process can occur too. A certain allele can be eliminated or diminished in a population through random genetic drift or disappear altogether. And that's why you have to have them fixed to make progress over time and build the genome, if you will. So let's add some more realism. This slide is going to overwhelm you a bit, and that's part of the point. Not to understand every detail per se, but to understand all the different layers of realism that come into play when you evaluate whether or not evolution is reasonably true. So we have to account for how long it takes for example, beneficial mutations to become fixed in a population. It takes a long time depending on certain circumstances. We have to account for random loss. Genetic drift is this random fluctuation of allele frequencies apart from selection, where just by having children you can lose those alleles just by chance. So you can lose that key beneficial mutation right after it arises. What is the ratio of good to bad mutations? Geneticists in the 1930s used to think, well, maybe it's 50-50. There's about, for every beneficial mutation, there's a harmful mutation. Is it 1 out of 1,000? Is it more like 1 out of 1,000,000? So what is the rate of beneficial mutations compared to bad mutations, or deleterious as they're called, deleterious mutations? What is the genome size? How much of the genome is junk versus functional? What about, are they diploid? Are they asexual? What is the mutation rate per nucleotide site? What is the population size, the number of reproducing members, the effective population size? Are the mutations occurring? Mutations always occur within linkage blocks. So you can't reshuffle individual nucleotides or DNA letters. Our genome is broken up into large blocks of nucleotides that are linked together. That affects the mutation selection fixation process. Has a profound effect on it. Selection interference. If you have one beneficial mutation, selection can compete for another mutation. Maybe it's a harmful one that has to get rid of. So selection is limited. It's not all powerful. Reproductive surplus. How many individuals in a population can you remove every generation through selection? Can you remove half the population? If you keep doing that, what happens? The population goes extinct. So there are limits to what the mutation selection fixation process can accomplish. What is a realistic distribution of mutational fitness effects? Like I said before, in the past people thought it was more like a normal distribution, with most mutations being either beneficial or harmful, or is it more exponential, with the vast majority being harmful or deleterious? So what is a realistic distribution of mutational fitness effects? And are most mutations selectable? In other words, do most mutations actually affect your reproductive success? Or are they too subtle? Are they essentially neutral? So what selection scheme is realistic? Is truncation selection? Is artificial selection? Or probability selection? Some people think that you can just rank everybody based on fitness in a population and eliminate all those below a threshold. That doesn't happen in nature. That's often used in evolutionary formulas when they use this analytical approach of testing evolution. So how do mutations interact? Is there linkage? Are those mutations linked together? How do they interact with each other in terms of improving fitness, like synergistic epistasis? What is the degree of dominance? Are these beneficial mutations completely dominant and therefore they're manifest right away, or are they recessive or have incomplete dominance? What about noise? There are many things that can interfere with selection and make selection operate on things that are not even genotypes. For example, if you're growing in a field, some plants may be more successful because they're exposed to more sun, and selection will operate on that, but not because it has a mutation. It has nothing to do with the genotype, it has to do with the environment. So things like heritability, is it high heritability or low heritability? Most traits have low heritability. All these things can interfere with selection efficacy, the effectiveness of selection. I know that's a lot, but all of these things need to be accounted for whenever you make a claim about evolution. But oftentimes I find people will speculate and say, oh, well, there's a strong selection pressure, and these apes living in the plains in Africa, they'll just evolve. It's overly simplistic. It doesn't account for any of these factors. Yet you see that time and time again being promoted by scientists even. So this is long, but I think this is important. This is from Dr. John Sanford and his colleague. Let's add genetic realism to Darwinism now. All those variables I just described, and some others, they combine to determine a population's fitness, whether it will increase or decrease. Unfortunately, there is no simple mathematical formula that can simultaneously account for all these variables simultaneously. So most population geneticists use what's called the analytical approach. It's very mathematical, and they have to use a lot of oversimplifying assumptions to make evolution work. In other words, it ignores biological realism. So as a general rule, the simplifying assumptions, as are required for a purely mathematical approach to population genetics, force researchers to ignore many variables that tend to reduce the efficiency of selection. This can result in overly simplistic expectations regarding the net fitness effect. Let me skip this, it gets a little in the weeds. Arguably, the only way to account for many biological variables that simultaneously affect the mutation selection process is by using comprehensive numerical simulation. So computers now have impressive computational power. You don't have to use oversimplifying formulas. And that's what Dr. Sanford and his colleagues came up with, was something called Mendel's accountant. It actually tracks mutations as they enter a population every generation and accounts for all of those variables, which is incredible but possible because we have such powerful computers now. So you don't have to guess as much and oversimplify. So as numerical simulations become more comprehensive and more realistic, net gain in fitness seems to become increasingly problematic. But that's the goal of evolution, increase in fitness, right? Through these novel adaptations. So here's Mendel's accountant. I'm not going to get too much into this, but I will just say that if you're interested in learning about this, this is from a creationist, he's a geneticist, but a successful decade for Mendel's accountant. He summarizes the past decade of research using Mendel's accountant, including the secular publications and creation-based publications that have been a result of this powerful program that models the mutation selection fixation process. So just to give you an example of how powerful this program is, it actually made a prediction that came true when they actually, so here's what happened. Who's heard of the 1918 Spanish flu? Very famous. It killed more people than died in World War I. So when it first emerged in the population, it was very devastating. And amazingly, Dr. Sanford and his colleagues, they predicted, using Mendel's accountant simulations, they predicted that this 1918 strain should be approaching extinction, if not already has gone extinct. Because just their simulations showed, as mutations accumulate linearly, fitness declines exponentially. And they said it's about the time where we would expect this to be going extinct. Incredibly and scarily, they actually have saved these genomes in the frozen, in the freezers, if you will, in the frozen fossil record. It's kind of scary, and they can revive them, and their genomes have been sequenced. And so what they have found is that, indeed, it went extinct in 2009. And I want to show you this incredible data. So they track mutations. Mutations accumulated linearly over time, and its pathogenicity, which is the best test of fitness. It's, how many people does it still kill? How lethal is it still? Pathogenicity declined in an exponential decay curve to the point where it went extinct in about 2009. Amazingly, Mendel's accountant predicted that, and it came true. And it was actually true in biological reality. And that was published in a secular journal. Highly accessed paper, has 25,000 downloads. So here's a Mendel simulation. So you can see mutation accumulates linearly, and then fitness declines exponentially, just like H1N1 in real life. So let me read this real quick. During the entire history of human H1N1, mutations accumulated in a perfectly linear fashion, exactly as seen in all our theoretical studies using Mendel's accountant. In the course of 90 years, almost 15% of the viral genome mutated, with mutation count increasing at a very constant rate. During this time, viral fitness, as reflected by associated human mortality rates, declined continuously and systematically from 1918 all the way to its apparent extinction in 2009. And I think that is science at its best. You can make specific predictions, and it bears out of the real data. So I will read this briefly. We have documented entropic genetic degeneration. In other words, declining fitness due to mutation accumulation within the human strain of the influenza virus that caused the Great Pandemic of 1918. Before examining the historical evidence of the genetic degeneration of the viral genome, preliminary numerical simulation experiments were done. So he's saying, look, we predicted this. Mendel's accountant was used to model the mutation accumulation within a hypothetical virus similar to the influenza virus. These initial numerical simulations strongly indicated that RNA viruses should rapidly accumulate deleterious mutations at a steady rate, causing the virus to run down and go extinct in a reasonably short time frame. On this basis, we predicted that this same pattern would be evident for the human H1N1 virus strain. Now, all that to say this, you can see why the field of population genetics, according to this evolutionary geneticist, the field of population genetics is now so well supported at the empirical level that the litmus test for any evolutionary hypothesis must be its consistency with fundamental population genetic principles. So if you want to evaluate whether or not evolution is true, is reasonable, is scientifically, has a scientific basis, you have to use the litmus test, which is population genetics. And how often do we see that? Almost never. So I want to do this. I think this will be fun because we're going to apply the principles of population genetics to a very acute scenario, a scenario that is well defined by evolutionists themselves, and we're going to test the ape to man hypothesis. So we know the generation time of chimps, right? It's close to ours. We know their genome. We have their genome sequence. We know their genome size. We know their population size, how large a populations tend to live in. We know their mutation rates, and we know the supposed divergence time from humans. So according to the evolutionary theory, human and chimps share a common ancestor. It's called the last common ancestor that supposedly lived about 6 million years ago. They don't have the bones of that individual, but they hope to find it still. And so they believe about 6 million years ago, you have this chimp-like ancestor, and one split goes to humans. The other split goes to chimpanzees and bonobos. That split happened 6 million years ago. That's the divergence time, according to the theory. So if humans and chimps, and I don't believe this is true, we'll talk about it, but if human and chimps are nearly identical genetically, 98 to 99% similar, it may seem very feasible, very easy for humans to have evolved from a chimp-like ancestor in the proposed time span of 6 million years. How many nucleotides would that be? How many mutations would that be if we're 99% similar? We'll discuss this. So we have to first, though, address this claim. Are human and chimps 98 to 99% similar? Well, a lot has changed over the years since that was first claimed and claimed again and again and again over the years. And I'm going to try to go through this quickly. You can read the sources more if you'd like. But the earlier studies omitted large segments of DNA. For example, when they were early on in genome sequencing, they would just compare the protein regions, which are the most similar between humans and chimps. They're the building blocks. Of course, they're going to be the most similar, but that's only 2 to 5% of the genome. So they ignored the rest of the genome, yet that's where the vast amount of differences are. So I'm going to breeze through this real quick. When you account for all the parts of the genome that were not considered, things like insertions or deletions called indels, those are large regions of the genome, the actual identity is much lower than what is reported in the primary scientific literature. And here's from Todd Prius. He is a primate evolutionist. In the Proceedings of the National Academy of Science, a very prestigious journal, he says it is now clear that the genetic differences between humans and chimpanzees are far more extensive than previously thought. Their genomes are not 98 or 99% identical. He says this here. Now, how similar are they? How similar are we? I'm sorry. It's very difficult to know, and here's why. In 2018, the journal Science published an article confirming what creationists have been saying for many years. We've been trying to say that, look, when you sequence the chimpanzee genome, you use the human genome as a scaffold to assemble it. And that created a very biased assembly, a very human biased assembly. And in 2018, they admit that the chimpanzee genome has been humanized. Their language, not ours. It's been humanized. Think about that. That means there's large parts of the chimpanzee genome that have been misassembled. So he says, most non-human primates' genomes generated to date have been humanized, owing to their many gaps and the reliance on guidance by the reference human genome. Precisely what I just explained to you. So here's a simplified view of this. Here we have the human genome. It was sequenced first. Not long after, you have short sequence reads of chimpanzee being brought up to the human genome. And wherever it would align, they would say, okay, here we have a match. What's the percent similarity? 100%. But in large areas where there was no alignment, like indels, they said, oh, no sequence alignment. We're going to throw that out, and we're only going to calculate the percent similar out of what aligned. The percentages were based on what was aligned. So, and because they used the human genome as a scaffold, in some cases they would deliberately put human DNA in the chimp genome as part of its assembly because they were sure that we were so similar, there's nothing wrong with this. There was deliberate contamination, and that's reported in their own journals. So it has been humanized. There is no doubt about it. It has been humanized. Now, the Y chromosome, the chimpanzee Y chromosome, amazingly, they said, let's forget about using the human genome as a scaffold to reconstruct it. Let's just reconstruct it based on long sequence reads of overlapping sequences and do it on its own merits. And guess how similar they found the human Y chromosome is to chimpanzees? 70% similar. And that's the chromosome that is the male sex chromosome. Most of that chromosome does not recombine with the X chromosome. The male-specific region does not recombine. That chromosome should be the most resistant to change over time because it doesn't cross over, it doesn't reshuffle. Yet, it was 70% similar to the human Y. And here's what they say in the paper, Journal of Nature. Indeed, at 6 million years of separation, humans and chimps, supposedly, right? The difference in the male-specific region, the largest part of the Y chromosome gene content, in chimpanzees and human is more comparable to the non-sex chromosomes, the autosomal gene content, in chickens and humans separated at what? 310 million years. So they're saying this is incredible. It's comparable to the divergence time that we would expect between humans and chickens. Those differences do rise. So evolutionary scientists, they're stunned by this too. They say, holy crap, and they use the same quote. And this is a popular human evolutionist who studies the ape-human fossils. So he's a paleoanthropologist, and they can see this does not fit what you would expect intuitively in an evolutionary model. Well, now they've actually redone the chimpanzee genome, and they have the latest version called PanTraw6, and it's been available since January 2018. And scientists have taken long reads of that, and they've compared it to the human genome, and guess what the similarity is overall? Not just the Y, but overall all the chromosomes. Well, two independent analyses using two different alignment software programs converged on almost identical results. So this is a former geneticist. I'm sorry, he's still a geneticist, but he was the former director of Clemson University's genomics laboratory, Jeff Tompkins. He's a creationist, so people will say his science doesn't count. I disagree. He's an impressive genomic scientist. He got 84% similarity of only counting alignable regions. Think about that. Only accounting the parts of the genome that align. There's still large parts that don't align at all, and those must be more different. So this is an upper limit. Here's an evolutionary biologist from Queen's Mary University of London, Richard Buggs. He says the percent of nucleotides in the human genome that had a 1-to-1 exact match in the chimpanzee genome was 84.3%. 4% had no alignment. So it's probably closer to more like 80%. Now, these are ballpark figures, and I won't say this is all gospel truth, but we are getting a sense that over time, when we have more clarity, chimps and humans are more different than they are similar. It's not going the other way. So let's just think about this scenario more accurately. That would result in about 500 million genetic letter differences between chimps and humans. But if we evolved from a chimp-like ancestor 6 million years ago, each lineage would accumulate its own set of mutations independently. So really, what we have to test for this, using this litmus test of population genetics to see if the ape-to-man scenario is feasible, you have to say, can a chimp-like ancestor evolve into humans when you need minimally 250 million mutations or nucleotide changes? So that's a lot of genetic differences that you have to account for in order to transform a chimp-like genome into humans in 6 million years. That's a lot of genetic information. But we're going to ignore that. We're going to assume chimpanzees are 99% identical, which is not true. And we're further going to assume that 90% of the human genome is junk. So it's only a 10% functional genome. So what you have to accomplish is really a best case scenario in this case. So at minimum, all we have to account for is about 1.5 million mutations. That's a lot of beneficial mutations that need to arise and become fixed in the population. You with me? So the problem, though, is this. Why is this such a problem? Because the mutation rate per genetic letter site, or nucleotide, is very low, 10 to the negative 8th. So it takes a very long time for a specific nucleotide to mutate into a specific other nucleotide. And 2/3 out of the time, it's going to be the wrong letter, because if it starts with a G, you need a T, it could be an A or a C. So the point is, it takes a very long time for a specific nucleotide to mutate. With this understanding, and in conjunction with this idea of fixation, how long it takes to spread to all members of the population, let's think about this more carefully, looking at evolutionary studies, including one study by Dr. Sanford, who is a former atheist. So here's Lynch, again, that very popular, very famous evolutionary geneticist I cited in the beginning. He did a study showing for just 2 specific beneficial mutations to arise in a model hominid population of 10,000. So according to evolution, we evolved from a effective population size of 10,000 individuals supposedly from Africa. And that supposedly happened, so that's the out of Africa model, okay? So we're assuming here a ideal hominid-sized population of 10,000. In modeling that scenario, they show that you'd have 2 nucleotides would arise, or 2 beneficial mutations would arise. It would take 200 million years, more than 200 million years. You see how problematic that can be. Lynch, again, 2010, over his other estimate, over 200 million years for just 2 specific beneficial mutations. Durrett from Cornell, 2 nucleotides, again, 216 million years. Dr. Sanford, he shows 84 to 270 million years using a different approach. These are analytical. This is using sometimes they use simulations too, but this is actually using Mendel's accountant, right in the ballpark. And he generously assumed each beneficial mutation had a fitness advantage of 10%. That doesn't happen in humans. That's like saying if I have a mutation, I can jump 10% higher, run 10% faster. It doesn't happen in human populations. So he's generously assuming in some of these a very unrealistically high beneficial fitness effect. Another study, 5 specific mutations, 200 million years. Durrett, this is amazing. Just one specific beneficial mutation to arise and become fixed, 6 million years. How long is that? That's how long we supposedly evolved since we evolved from the chimp-like ancestor. But yet we're talking about how many more nucleotides do you have to change a chimp-like genome into man? Many hundreds of millions. 65 billion for 8. 65 billion. We'll talk about that more. Why is it so hard for this to become a reality? It's because unlike harmful mutations or deleterious mutations that are harmful can arise anywhere in the genome. There's no specificity. It's easier to break something than it is to fix something. But to fix something, to improve it, requires specificity, right? And if you have to have a lot of mutations, or a few mutations even, those have to be coordinated. So that's what evolution has to accomplish. To change a chimp-like brain into a human brain with Broca's speech area, or the ape foot to a human foot, those are many coordinated mutations. They can't just arise anywhere in the genome. They require specificity. So that's what they're modeling. And in this scenario, Dr. Sanford is he's assuming that you already have a pre-existing gene that was duplicated. A best case scenario. You don't have to start from scratch or de novo gene birth. You can start with a pre-existing gene, duplicate it, and all you need are, say, 2 mutations to arise, and then it becomes functional. It becomes a novel function or structure, and then it can be amplified or has a fitness advantage where it can be amplified by selection. Does that make sense? So he's assuming a best case scenario. You already have most of it already finished serendipitously, and you just need a couple to complete that novel gene. Am I talking too quickly? Okay. So to wrap up this part, and we're almost done, to transform a chimp-like creature into man over a period of 6 million years, at best you could maybe do 2, 3 specifically placed mutations that will arise and become fixed. Keep in mind, that results in the most trivial type of change you can imagine, resulting in maybe a single new protein fold. That is very unimpressive. Here's a plot from Dr. Sanford's published paper, again, a secular paper, and he shows that as the number of beneficial mutations, specific mutations, are increased, the waiting time to fixation increases exponentially. So here you have, for just 8 nucleotides, they said 65 billion years, he says 18 billion because he's using more generous parameters. For example, he assumes that you can eliminate 50% of the population each generation and still maintain a stable population. He's being very generous, and he's still getting it exceeds the supposed age of the universe according to the Big Bang. I think you're beginning to see that as you add more and more layers of realism to Darwinism through population genetics, it becomes insurmountable to make even the most basic changes, at least in larger genomes with longer generation times like in large mammals, humans, elephants, whales. So, testing the ape-to-man hypothesis. Here are some of the references. You can screenshot that, or I can give that to you later. And here's Dr. Sanford's paper on this topic, published in 2015. This paper is very profound, and I recommend everyone to read it. If you are a biologist especially, I highly recommend reading this, learning it, studying it, because it really takes into account genetic realism. So, why does it take so long? I want to explain to you one of the reasons why it takes so long. First of all, we know it's because, well, beneficial mutations by definition are already very rare. And already we know it takes a very long time for a specific nucleotide to mutate into a specific other one. But it's also because of something called genetic drift. So let me just discuss this. This is according to population genetic theory. This is not something that creationists are making up here. So if you have a fitness benefit of, say, 10%, that's highly unrealistic, like I said. And evolutionists understand that. And they think even this, 1%, is strongly beneficial and is not very common. Evolutionary population geneticists think that a fitness increase assigned to a single beneficial mutation is more like 0.01%. Here's the problem, though. Even if you assume a strongly beneficial mutation, very strongly beneficial, that mutation in that exact position has to arise in the genome 100 times over and over and over again in order to escape being lost due to random genetic drift. Remember, when a mutation first arises in a population, it's vastly outnumbered by the non-mutant allele, the wild-type allele. 1 out of 20,000 in a human population of 10,000. So, because it's so rare initially, it's more than likely going to be lost from genetic drift while it's still a rare allele just by chance. In fact, over 90% are lost right after they arise in a population. So because of this known, well-established fact in population genetics, a single strongly beneficial mutation has to arise 100 times over before it can finally persist in the population. But that's just the first step. After that, remember, it has to spread to all members of the population in both chromosomes. Can you see that? You see how long that would take when we're waiting for these rare beneficial mutations to arise, overcome loss to drift, then go on the long journey to fixation. And larger population sizes mean longer time to fixation. So any way you look at this, there are trade-offs. Okay, so let me read this from Durrett and Schmidt. This is, again, evolutionary population genesis from Cornell. They say, first, we have assumed that once the target word, in other words, once that novel functional gene occurs in an individual, it sweeps to fixation in the population right away. They're saying that's not true. That can't be realistic. They're admitting that. That is, not long after introducing this frequency will rise to 100%, right? In reality, the probability of fixation is approximately the selective advantage conferred by the mutation's selection coefficient. And even for strongly beneficial mutations, we have a selection coefficient of 0.01 or 1% fitness gain. This is the key. This means that the mutation would need to rise more than 100 times in order to achieve fixation, which would increase the waiting time. So again, I'm not making this up. This is accurate, and it's from their own papers. So we must multiply all of their estimates in his paper by 100, and they freely acknowledge that and understand that. But they write that at the end of their paper. So when they say 650 million years for a sequence for 8 specific beneficial mutations, that actually takes 65 billion years. So that's just one reason why it takes a very long time for mutations to arise and become fixed. It's because genetic drift, random loss to genetic drift, is one of the most significant rate-limiting factors. And here is Michael Lynch, again, very famous evolutionary population geneticist, in a major journal. He writes, a central problem in evolutionary theory concerns the mechanisms by which adaptations requiring multiple mutations emerge in natural populations. Can you think of a scenario where we would need multiple mutations arising? The ape-to-man scenario. Most any macroevolutionary scenario would require that. But again, it's most problematic in larger genomes, longer generation times. And so this is actually another key paper that shows they're actually trying to resolve the waiting time problem. But in doing so, as I showed you, they still confirm the reality of the problem, despite their many unrealistic assumptions to make the scenario more likely for evolution. So this is something that I wrote, I'm summarizing. Applying the litmus test of fundamental population genetic principles effectively falsifies the ape-to-man hypothesis. The genetic transformation of a chimp-like ancestor into man would minimally require 100 million nucleotide substitutions, beneficial mutations, and they have to become fixed. Yet the waiting time for just 2 codependent mutations to rise and become fixed in a model hominid population of, say, 10,000 is on the order of 80 to 200 million years, according to a creationist, former evolutionist, and evolutionary population geneticists themselves, simulating comparable scenarios, 10,000 population size, approximately the same mutation rate, and so on. How then can the touted macroevolutionary transformation of a chimp-like ancestor into humans be considered genetically feasible in any amount of time? And I haven't even talked about something called orphan genes, which means you cannot take a pre-existing gene and duplicate it because it has no similar precursors. And so you have to essentially start from scratch. And that means it's called de novo gene birth. So that alone defeats the ape-to-man scenario, that alone. So here's the devastating flip side, and I'm going to close in a couple more slides on these key points here. Here's the devastating flip side. So far, we've only considered beneficial mutations and the long waiting time for them to arise and become fixed. I haven't even talked about harmful mutations or deleterious mutations. This makes this incredibly more difficult. So we need to add another layer of population genetic realism to Darwinism. So far, we've only considered the waiting time for fixing rare beneficial mutations in a population. But we must also consider the devastating effects of fixing the most prevalent class of mutation known as deleterious mutations. And here's from a population geneticist. The vast majority of mutations are deleterious. This is one of the most well-established principles of evolutionary genetics, supported by both molecular and quantitative data. So here's the point I'm trying to make with this. While waiting for a few very rare beneficial mutations to arise, to escape random loss to drift, and then increase in frequency to become fixed, vastly more deleterious mutations will accumulate in that time period. They will escape loss, and they'll become fixed in the population, representing irreversible genetic damage. So good luck waiting all that time for those few key beneficial mutations. Meanwhile, the rest of the genome is degenerating. Now, this problem is mostly a problem for long timescales, evolutionary timescales. That's important to understand. So objection. Wouldn't most deleterious mutations be removed from the population due to purifying selection or negative selection? If they decrease an organism's fitness, they're less likely to reproduce, right? So why can't they just be eliminated? That's the knee-jerk response. And for that, I have about 50 more slides to address it. But I'm not going to do that to you. I'm not going to torture you. Just let me summarize what's ahead, a sneak preview. The heart of the issue is this. That can only be true if most mutations that are harmful are selectable. Think about that. In the past, for example, since Ronald Fisher, he assumed that most mutations, it's equal, 50-50. Beneficial and deleterious are equal. But actually, a more realistic distribution is more exponential. That means it's a gamma distribution. That means the vast majority of all deleterious mutations are very close to zero fitness. What does that mean? It means they're called nearly neutral, population geneticists call them, or very slightly deleterious mutations. In fact, they say there is no such thing as truly neutral mutations. It's not true. Even synonymous mutations affect the speed of translation and protein folding, and they say they're more accurately termed nearly neutral because they still alter the genome, they still cause fitness decline, it's just very imperceptibly small. But the problem is this though. If the distribution of fitness effects is closer to the red, exponential, with a vast majority of mutations being very slightly deleterious, then no purifying selection can stop degeneration. Why? Because they're invisible to purifying selection. They're below the selection threshold, so they accumulate in the genome without being selected away. They act, and so they respond to natural drift, to genetic drift, not to selection. And here's a geneticist whose famous paper says contamination of the genome by very slightly deleterious mutations. Why have we not died 100 times over? So let me just do a summary of take-home points. There's a lot more I could say about that. And I think the second half could honestly be just as devastating as the first. The more layers of realism you add to Darwinism, the more you see this is not good science to claim evolution is a fact. Beneficial mutations require specificity, right? That makes sense to improve things. Deleterious mutations cause damage wherever they arise. Deleterious mutations vastly outnumber rare beneficials by about 1 to 1 million, according to Richard Lenski or other population geneticists. While waiting for rare beneficial mutations to become fixed, the rest of the genome is degenerating. Because these very slightly deleterious mutations become fixed at a rate equal to the neutral mutation rate. So if the neutral mutation rate, let's say, is let's say 2 or 10 or 50, that means every generation, 50 become fixed every generation. And they get fixed through genetic drift, and so selection does not operate on those. Very slightly deleterious mutations are the most prevalent class of mutations. Yet they are invisible to purifying selection, I just said this, and become fixed via random genetic drift, resulting in perpetual fitness decline. Over evolutionary timescales, this is a devastating problem. As a beneficial mutation goes to fixation, many more deleterious mutations hitchhike with them on large linkage blocks. I didn't explain this yet. I'll do it briefly. As I mentioned, though, I did mention this, nucleotides at the individual level don't reshuffle during recombination, for those who are biologists. They exist in large linkage blocks. So it's kind of like this. Imagine shuffling a deck of cards, but gluing most of the cards together. Those aren't going to reshuffle. They're linked together. So even when you have this rare beneficial mutation that is highly selectable and it goes to fixation, guess what? It also brings to fixation all the far more numerous deleterious mutations with it. So the net effect in fitness is down when you change the environment to a new situation. So this is called Mueller's ratchet, essentially Mueller's ratchet. So even when you assume beneficials are going to fixation, it's not helping. So population geneticists admit if the mutation rate was as low as 0.5 mutations per person per generation, it would be devastating. The actual mutation rate is 200-fold higher, and the junk genome paradigm has collapsed. So that's about it for now. I hope that you don't feel like I sprayed you all with a large water hose of information. I feel self-conscious about that, but this is more resources. I did a book with Dr. Sanford. We did research on, I didn't even share my research on using Mendel's accountant. Maybe another time I could share a paper I did with Dr. Sanford on that. Has to do with fixation rates again. But I also worked on a book with him for several years, and we wrote a book called Contested Bones. And it focuses not just on genetics, we do that in a chapter, but we also focus on the so-called ape-human missing links, like Lucy, which is Australopithecus afarensis, Australopithecus sediba, Homo floresiensis, Homo erectus. You've probably heard of those, Neanderthals, Denisovans. Genetic Entropy, this is one of my favorite books. I'm biased because I worked with him for many years, but this is an excellent book if you want to understand more about population genetics and all these different layers of realism. And then finally, this is a DVD I came out with in 2020. Scene 4 is free on YouTube. We talk about genetics of human origins. How do you explain the different people groups? Is it true we came from Adam and Eve? Well, how do you explain how we could all come from Adam and Eve if there's a lot of genetic variation in the population? Things like that come to mind. There's incredible evidence that all humans can be traced back to a single father and mother of the human race, and I don't think that fits with the out of Africa theory as is often claimed. So you can check out that if you'd like. You can stream it on Amazon, or Scene 4 is free on YouTube. There are videos trying to challenge it, but I haven't had a chance to respond to those. I hope to, because I think the answers are pretty easy to rebut. And then back to Genesis, this is the nonprofit I have. I recently published an article refuting critics' claims against genetic entropy. So that has to do with genetic drift, and it's a little, not too technical. That's it. This is the small nonprofit I have on the side. Mostly now I'm focused on geology, though, so I've tried to just focus mostly on exciting things like sand and mud. And then Logos Research Associates, go to logosresearchassociates.org if you want to help fund creation-based research. And I'm actually raising funds for part 2. And I want to do a film, hopefully, on population genetics versus evolution as well, and get this out to a larger group of people. So that's all. I really apologize for taking up so much of your time, and thank you so much. Really appreciate it.